Okay, one more lecture here on viruses, and we're also going to talk a little bit more about prions here. Okay, your textbook has a section where it talks about cytopathic effects of viruses on their host cells, so we can kind of figure out what this word means by breaking it down. You guys know cyto refers to a cell, like a cyte, C-Y-T-E. Pathic, like pathogenesis, you're making a cell sick. So viruses often, or typically, make their host cells sick. Because remember, a virus is just trying to get itself replicated. It doesn't really care whether or not its host cell survives. And so very often viruses do trash their host cells. Now we've already talked about how many viruses cause their host cells to lyse or to burst so that the new virus particles can actually exit and go floating off to find a new host. There are some enveloped viruses that do this weird thing where they cause the host cells to fuse together and form giant cells. Those are also called syncytia, and that's what you're seeing here in this picture from your textbook. Right in here, all the light pink, those are big giant cells, and in dark purple, each of those is a nucleus. So that's letting us know that these host cells fuse together and made these big giant cells. And the reason they do that is because, let's say, it's because of budding. All right, so if you got a host cell here, there's a host cell, and you got a virus there that's budding, and that budding virus can attach to a new host cell over here that's adjacent. And when it does that, it can actually cause the cell membranes to fuse or melt together. And now you have a bigger cell that has two nuclei on the inside. And then you can imagine if you've got a vigorously replicating virus where you've got all these new viruses budding and they're reaching out and attaching to other new host cells, pretty soon you can wind up with these big giant cells that are called syncytia. Many cause their host cells to round up. And I think we've got some example pictures of that over here. Yeah, this is, if you were, all right, if you grow virus, we cannot grow viruses in our lab, as I've told you before. Well, actually, we could in our lab over in Huntsville, but we'd have to go into the biotechnology class portions of the lab. And it's a lot more complicated to grow viruses because you have to grow them in host cells. So typically those host cells are grown in little containers like that. That's called a flask. And they're growing in a sheet on one surface of the flask, and that red fluid you see there is the medium that's providing the nutrients for those host cells. So a lot of times those are human cells of some type that the virus will use as its host. And if you look at those host cells under the microscope, you'll see something like this where they kind of look like tiles. You know, each of those is an individual host cell. And if you infect those with the virus, many viruses will cause those nice, pretty cells that are all laid out like little tiles or sheets on the surface of a flask. It will cause them to look like this. They round up and look really, really sick. And that's how you know that there's a viral infection going on inside that flask of those host cells. So kind of the bottom line is that viruses are not always nice to their host cells. All right, how do we detect viruses for diagnostic purposes? So traditionally, we haven't been too good with this, and you guys know that. When you go to the doctor thinks you have a virus. That's all you find out. You don't know exactly what kind of virus you have many times. Now, we do have tests for the flu, and we have tests for other types of viruses now as well, like human papillomavirus, HPV, and HIV. But a lot of times you don't know what you have. That's beginning to change. We now have this PCR technique that I mentioned very briefly on Wednesday. That is a genetics-based technique that can very specifically identify the types of viruses that you're infected with. And it has many other applications as well. That's not the only one. Then there are what we call immunodiagnostic assays. Toward the end of the semester, we'll talk about antibodies. Those are weapons of the immune system, naturally made by the immune system, that attach to things that are foreign to our bodies. they're so specific about what they attach to that you can actually use them in the lab as tools to help you detect things. And so when you do that, that's called an immunodiagnostic assay. You're diagnosing something by using these antibodies of the immune system. So they're very sensitive. Many different examples of those types of assays. We'll do blood typing in our lab before the end of the semester. We will probably do home pregnancy tests as well and talk about how those work. No, that doesn't mean you personally are going to be doing a home pregnancy test. You could if you wanted to. They are real home pregnancy tests, but I've never had a student choose to do that. Those work this way. Those are immunodiagnostic assays. Or if you've ever had a rapid strep test, or if your kids had a rapid strep test at the doctor's office, those work pretty quick and they're very sensitive, and that's also an immunodiagnostic type of assay. We'll see some examples of those a little bit later in the semester. Viruses, typically you can't view them with a light microscope. You can't grow them on agar or in broths like we do with bacteria in the lab. So it's more expensive. The people who grow them have a much higher level of technical training, and so traditionally we haven't been very good at diagnosing what specific virus you're infected with, but that is changing as we move through the future. There are some viruses that cause cancer, and your textbook has a little bit of information about this. Again, cancer cells are cells that are dividing in an out of control manner in your body. Some viruses have genes that are called oncogenes. Onco refers to cancer, like oncology. An oncologist is a cancer doctor. And oncogenes are genes that code for proteins that can trigger cells to become cancerous. Okay, and sometimes viruses have their own oncogenes, or sometimes they can turn on genes inside our cells that trigger those cells to become cancerous. Not all viruses do this. It's restricted to certain types, but some good examples are human papillomavirus, or HPV. We've all heard about that because there's a vaccine for that now to prevent cervical cancer. Epstein-Barr virus, or EBV, that's the mononucleosis virus. It infects B cells, B lymphocytes, a type of white blood cell, and in some rare cases it can actually trigger a B cell to become cancerous, and you can develop something called B cell lymphoma. No, that doesn't mean if you had mononucleosis you're definitely going to get B cell lymphoma, but you do have some risk of that happening. Just like if you've ever had genital warts, human papillomavirus, that does not mean you're going to get cervical cancer, but you are at some risk of that happening. There's another cancer-causing virus, human T lymphotrophic virus type 1. That's kind of a mouthful. It's also called HTLV1, and it is a relative of HIV. It does not cause AIDS, but instead it can cause T-cell cancers, types of leukemia in some cases. All right, now what about these prions? So we talked about prions in Unit 2 because we mentioned that prions are actually the most difficult infectious agents to control. They're up at the top of the list. They're almost impossible to inactivate. So what in the world are these things? They're very strange because they are infectious proteins. They have no DNA, they have no RNA, no genetic material. They're not viruses, they're not bacteria, they're not any kind of living thing. They are simply a protein that is capable of multiplying and spreading and damaging brain tissue and causing death. There's no treatment for these types of diseases yet. So this is really strange. So when it was first proposed, I think back in the 1980s, that these prions existed and they were just infectious proteins. Nobody believed it. It took a while before this was actually proven. Some examples of prion diseases include scrapie in sheep. Mad cow disease is the thing that we have heard about. In humans, there's a version called Creutzfeldt-Jakob's disease, or CJD, which you may hear about. That's also a prion type disease as well. The clinical term for the diseases these cause is spongiform encephalopathy. Spongiform, like the brain becomes spongy due to these diseases. Encephalopathy is referring to pathogenesis, disease within the brain. And over here, this is a image of some brain tissue from a brain that was infected with prions. And in pink, those are living cells, mostly neurons that took up a pink dye because they were still alive. And in white there, all those holes you see, those are places where neurons used to exist, but they died because of these prions. All right, so what are these prions? They're kind of strange. They're actually derived from normal neuron proteins. The normal function, though, is not well understood. The prions are a misfolded version, kind of like a partially denatured version of some normal neuron protein. And if that's not bad enough, they're able to convert other copies of the normal protein into the prion version. So they function like an enzyme. And then those newly converted prion proteins can turn other normal proteins into prion proteins. So this is a protein-protein spread. There's no DNA or RNA, no viral replication, none of that going on. It's simply a protein. It's kind of like a zombie. These are like zombie proteins that are able to find other normal members of its population and convert those over to the prion or zombie form. Let me skip over here to this diagram, just kind of give you a visual on that. All right, so prion diseases are caused when you've got some normal neuron protein. And again, we still don't know what these proteins naturally, they have some natural function in the nervous system, but we don't know what it is. They mutate and become the prion form here. So it's misshapen. And then these prion forms act as enzymes and convert other normal versions of the protein into the prion form. So again, they're kind of functioning like zombies, and then this spreads across neurons in the brain. And this is a diagram that is showing you that as well. This is supposed to be a neuron here in blue, and the little green spots you see there, those are the normal proteins, which is also called PRP. And in orange, those are the prion versions of the proteins, the misfolded ones. And the orange ones find other green ones and convert them into the prion version, the orange version of the protein. And what happens is these prion versions all clump up. They form big clumps inside the neurons, and they accumulate, and eventually the cells die. And neurons don't get replaced. So once a neuron dies, it's dead, and you can't replace it. And so eventually you wind up with these holes in your brain tissue, and the brain becomes spongy, around the time of death, which is where that term spongiform or spongiform encephalopathy came from. All right, one thing I want to point out, okay, so these are normal proteins in the nervous system. So they're proteins. So there's a gene on your DNA that codes for those. So a few minutes ago I mentioned Creutzfeldt-Jakob's disease, or CJD. That is a disease that occurs spontaneously in humans. Okay, and what that means is the gene, there's a neuron somewhere, and the gene that is coding for this normal protein spontaneously mutates, and now it's coding for the prion version. And once that happens, those prion proteins, again, they're like zombies. They're going to go around and find other normal versions of the protein and convert those into the zombie prion versions. That's called CJD. You don't get that by eating some kind of food. You just get it because it spontaneously happens due to a genetic mutation in the brain. So you may encounter during your careers patients who have CJD. Now, you have to be very careful handling the brain tissue of somebody who has CJD, because if you wound up absorbing those CJD prions by handling, like during an autopsy or something, if they got into your bloodstream, they could spread to your brain, and then you could wind up with this illness as well. Pretty unusual, though, but there are some cases of it. Mad cow disease, the reason people can get that is because if you have cattle who have mad cow disease, they have these prions in their brain. And unfortunately, that can develop because cows at times are fed the body parts, including the brains of other cows. So if it's unknown that there's a mad cow disease outbreak in a cow population, it can spread by cattle feeding off of the brain tissue of other cows who have these prions. But anyway, if you eat beef that's contaminated with these prions can actually get absorbed through your intestinal tract into your blood, and they'll travel, they can pass across the blood-brain barrier and get into your brain, and then you can develop mad cow disease from that. Very rare, that's not very unlikely to ever happen to you, or you're probably never going to see a case of mad cow disease, hopefully. But just be aware that it's out there, and do keep in mind that every once in a while there are people who spontaneously develop these prions to do a genetic mutation in their brain, and that's called CJD. Very interesting subject. If any of you guys have questions about it, please bring them to class when we meet on Wednesday so we can talk about it some more. All right, so that finishes up our discussion on viruses and prions, and the next video clip I'm going to start on the chapter 10 material, which is about antimicrobial drugs, chemicals that we actually give to people to try to treat infectious disease.